Alright, so this will be the first video on this channel. I am using Cell Lab on my phone, Android device. It is a modded version. It is Enzyme. It is a more performant version that I have uploaded in the past on the server. So, in this video I will vaguely go over simulating Hox zones or Hox domains, a form of gene expression within especially embryonic phases. It is how animals and such form segments along their body, or arms, legs, heads, dedicated organs, such and such. In Cell Lab, we can mimic that either by going extremely advanced and mimicking DNA itself, like many have, and it's very impressive. I do not do that. Or you can just use signals, because just like in real life, in Cell Lab, for every adhesion that a signal passes through, it pretty much exponentially decreases very rapidly. So you can use greater than and less than statements within stem cells in order to decide, based on how far away a stem cell is from a hormone or a signal, what it becomes. So it kind of generates a circle, a radius around, let's say, a secreting cell, secreting signal 1, or a radius around like a sensocyte or something. So you could even have a embryo of sorts develop depending on how much light is around it. So it might create more photocytes within its body in its early stages if there is less light, so that it can grow faster. Things like that. So there's a lot we can do with it. It is very fascinating, and I have experimented with it quite a lot, mostly inspired by Aboba, who is to my knowledge, Russian creator, who was using this similar method to generate complex brains, or attempt to. He succeeded and not succeeded. There really is no failure in this. It's all experimentation. What he has done is very impressive, and I built upon it, specifically aiming for body plan structure versus what he was doing, which was compact neural structures, which I am very uninformed in and not good at. So no, I cannot describe much of that to folks, at least in Cell Lab simulations. But here, I can, in fact, show you, and I do have the right one, show you this one. So this is a creation I made using this. We will observe, and then we will freeze it. So, this is an organism I made. This is it during its embryonic development. It creates a ring that it then gets filled with stem cells. So this relies on the fact that this is a basic mathematic principle observed everywhere in life, in the real world, that a bunch of circles being packed together will form a hexagonal or more crystalline structure. This is how crystals form, how you get cracks and stuff, and you can even see it here. We have some cracks in the structure, but for the most part, it is a hexagonal structure. So the way we do this in Cell Lab is you just produce a lot of free-floating cells within a cavity. They will automatically shift into place depending on the size of the cell and the size of the cavity. And then what I prefer to do is have all of the stem cells turn into gluriocytes, so that they, as you can see in the middle here, start to bind to each other. And then those gluriocytes will almost instantaneously split. And when they do, it's like 90% priority on one cell, and then the other daughter has 10%, so it dies immediately, and it preserves all of its adhesions. So it allows you to very quickly, using just a couple of modes, snap everything together, adhere everything together, and form strong connections. This does add a little bit of randomness. As we watch, it will constrict together, as now the red will turn into gluriocytes. They will link together, and then shortly they will grow enough mass to then become stem cells. and we can start seeing that propagation. The green gluriocytes will also turn into photocytes to help give some energy production to this organism. And very rapidly we can see this. So, of course I hit the wrong button. It's fine that it did that. As we can see, all of the green stayed photocytes. That's how I designed this. It creates this kind of square pattern where you get one in each corner, and then you get a clump in the middle. It varies every time, just a little bit, but this is primarily what it looks like. And then all of the blue, so the lipocytes, and these are flagella up front, and these stem cells back here, all of these were the red stem cells that we saw. If we clear, because that was technically an accident, I am not going to pretend it wasn't. We can observe again, and slow observe. Well, we will try to... There you go. It's very hard in Cell Lab because you only get 2 speeds to try to use. So I will try to explain what's happening as it's happening. So all of our red gliocytes will transform into these red stem cells. And upon doing so, most of them have. I'm not going to wait for every single one. What we see happens, I'll even show you the programming. So load from genome, or load from microscope. This is number 18. This is a 50-mode organism. So we're not very far yet into the organism's genome. Honestly, almost a third of the genome is just the tail in this design. So this actual self-packing mechanism only takes a few modes. So that's quite handy. What we can see with this stemocyte, which is very crucial, is in its tree down here, its paths. This is a only 2-path stem cell. I believe there are mods out that add like 4, or at least prototype ones that have become lost to the archives. But 2 works just fine. So as we can see, all this is doing a greater than or less than equal to. That's all. That's one of the values that Cell Lab can do. And we are looking at C 0.29. So number 18 is checking to see if S16, in this case, it could be S1. I just chose 16 because it's far away from all the others. S16 is being produced by, if we go back to our microscope, this, let me just climb our way, this secretion cell here. So as the secretion, as signal 16 travels through adhesions, it gets weaker and weaker and weaker until by the time it reaches down here, we can probably actually witness it in diagnostics. We can't because it does not scroll down to 16. That's kind of funny. Well, I can promise you 16 says 0, just like everything else. Kind of odd that it's not showing 16. I think it's because I'm in horizontal mode, because it usually will. So, you get this natural gradient where signal 16 is very strong towards, the closer you are to the secretion cell, and then it gets weaker and weaker in a gradient through the body. You could, in theory, add a secretion cell at the bottom, and maybe even one on the left and the right, all producing different hormones to create four overlapping radiuses or circles of signals. So you could force the organism to have a left side, a right side, a back, and a front. You could go even further. If you made like a long snake-like organism, you could have, if you're using this mod, up to 16 of these signal producers, all with their own radiuses, so that each section of the organism, using the same tree of stem cell decisions, make it have different organs and things like that. And this is what I used in my big cell, as I named it. I did not give it a fancy Latin name or anything. It's just called big cell. I've uploaded that before, and I will make a separate video on it. That uses that premise, or a similar premise. So we will show that off at some point. But going back to the genome, as we saw with it, if it is above 0.29, if it is a large number, so like one adhesion or two connections away, this will become flagellum. Otherwise, it'll become stereocyte number 22. Pretty easy. And 22 has two decisions, just like the previous, using the exact same principle. But now it's looking for only, as you can see, 0.04. Again, the drop-off in signal strength is exponential, so you can really only get about 4 distinct rings around a secretion cell, just because of the size of cells in Cell Lab and how fast it diminishes. I really do wish we could adjust that value in settings, or per organism, like the strength of a chemical or something, but Peter never did that, so we don't have that function. So we have to put up with these numbers. I had to tweak these numbers a lot to get it the way I like. You do not have to use these numbers. You can add way more bias. You can have 1 zone, 2 zones, 3, 4. Maybe if you use absolute cell values, you can tweak it to get 5, but I doubt it will act very consistently at 5. Or, like I said earlier, you can just add another secretion cell at the back of the organism and have it form that. Another thing you could do as well is one of these zones could turn into secretion cells to make a new radius of zones dictated by the first radius of zones. And that's more accurate to real embryonic development of hormones and proteins and such, creating a radius, a gradient of concentration in stem cells reacting to it. Stem cells will become stem cells that, will produce their own chemicals in order to signal its neighbors. It's a very amazing cascading effect that you can have occur in these processes, and I love it. Before I nerd out too much, we will continue. So M22 will either become the lipocytes, so that's our second ring that you saw in the organism, and then the other option is M24. We'll look at 24. 24 is the egg. So I have designed 24 to be an egg of sorts that upon we can even look, it only has one decision. This is age-based. So I calibrated this age specifically for this organism based on the times that, or the ages of the outer shell and when the shell dies off. So it relies on the adult dying for the eggs to develop into adults, which is not... I can't name any off my head, but I can almost guarantee there are real examples of organisms that just hold eggs in their body until they die, and then when the parent dies, the eggs just release or hatch within and eat from within. I think that I've seen it happen in tardigrades, like a dead parent that has babies within it eating from within. But I don't think it's like that on purpose. That was just a one-time event I saw online. So it can happen. But this is designed to do that on purpose. So we will continue slow observing, because it happens fast enough that even in slow observe, this happens pretty quick. So we should see these front cells become flagellum, which you are starting to witness, because they are close to the secretion cell. They are roughly facing forward. Some face a little left, some face a little right, so each of these little algae I created will kind of drift one side or the other better than the other. And we should start seeing these middle ones switch to the next one. So these are switching to M22, which then decide to become lipocytes. Or M, I believe, 24 or whatever it was, to the blue. We will also see the outer shell makes its own decision, where the front have become different from the back. So we get some differentiation within the shell too, which is always very interesting. So now you can see we have formed our 3 layers based off of where this secretion cell is. And they are 3 unique layers. In this case, we got a lot of eggs. Sometimes you only get like 6 or 8 down here. This one, because of how the adhesions happened, have created a lot. Sometimes at the end here, at the base here, where these gray keratin are formed, sometimes you get 5, like we see here. Sometimes you get 6. I've seen it even have none. Sometimes the adhesions just happen in a way where tails never form. But while that may not be fully realistic, you will get organisms that differentiate a lot like that, like how many flagellum they have and things like that. You can even see it within like sperm, like dual tails or single tails. So more simple life, like single cell and stuff like that. But in this case, in the design that I've made, there is quite a lot of variables. You can see a lot happen within one organism. If we go back to our genome and just load our guy again, you get to appreciate my bad organism names before they actually get an official name. And we sterilize the plate. We can see just how fast this development happens in real time. Just to show off that this decision making with stem cells is very rapid. Stem cells are very, very fast at their decisions. And as you can see, bam. Under 10 seconds and this organism is off and swimming and doing its thing. Isn't that pretty cool? I think it's pretty cool. And we can see it working. He is going to avoid this wall. This design happens to actually be pretty decent at avoiding. And I'm pretty proud of that. Before I end this video, because this organism I'm about to show off is unfinished, but it was going to follow similar principles, as you can see with the name 12 Hox Gene Expression Domains. I was experimenting with a similar concept, but for length. So without using a number of splits or max number of splits, instead these center stem cells are detecting the signal created by these 2 secretion cells, and that's how they decide to stop. So the length of this organism is depicted by those inner stem cells and how reactive they are. Nothing else happens in this organism. Again, I didn't program it fully yet, but we can see a similar thing. I have an empty cavity that connected to itself, so you get this hexagonal packing pattern that can create some pretty complex shapes. A bunch of algae in the middle to make it nice and bright green. You could, for example, make all these cells like buoyancy cells that are colored white, so it kind of looks translucent to mimic like cytoplasm or something like that, if you wanted a mostly transparent organism. But more realistically, what I was planning to do is have the outer shell have multiple zones, have a tail and a head. So this would be like, stem cell 15, but these could also be stem cell 15 and just make the decision based off of what hormone they are detecting. And automatically decide where the head is versus where the tail is. So this method allows you in a much smaller amount of modes to dictate like orientation, like what orientation is the organism, and how the organisms form. So these secretion cells are creating that 4-layer, roughly to the middle, 4 zones, and then this guy's also making 4 zones. So that alone gives you 8 distinct zones within this cavity that we're looking at, that I could program this one mode, this is just one mode, to make decision A or decision B. And then the next one, it'll say, okay, now make decision A, decision B. And with 8, it should only take 2 to 3 decisions based on how you do it to get all 8 of those zones to be distinct cell types. So we could make a similar organism with flagella up front, lipocytes, eggs in the back, Sadly, there's not a lot of cell types in Cell Lab, so it's very difficult to properly make internal organs because cells in Cell Lab are quite universal. You only have a couple energy producing cells, and photocytes are OP. So at the end of the day, it's kind of hard to make interesting organs or organelles within a organism in this simulator. But you could mimic them if you'd like, or try to. You could make, so like I said, eggs in the back. Maybe next to the eggs are like pulsating muscles to mimic like like gills that ripple. So you can mimic with movement a organism that is breathing or something like that. If you had some secretion cells on the left and right, you could have muscles form in the middle that are left and right specific. And in this instance, I believe my goal was to have a brain of sorts develop in the front. But I haven't gotten that far with this one. This is kind of a dead project because I took a break from this one to work on the big cell that I made a TikTok and Red Note about. So since then, I have not worked on this project. Maybe I will come back to it. Who knows? But I know this wasn't much of a tutorial. This is more of a discussion of what can be done in Cell Lab. This has 0 editing, 0 filtering, and we've been sitting here so long that the organism's about to start dying of old age, which is fun to watch, so it's a perfect time to stop the video. But I hope you enjoyed. For any of the Cell Lab like technical players that prefer these more complex topics, I hope this was useful. I hope for even new players this showed you that there are more methods in Cell Lab, or really any cell simulator that has hormones and stem cells, to allow the physics engine to build the body for you. You don't have to sit in your genome editor for hours upon hours sculpting a very hard to keep track of and sophisticated body shape. You can allow the physics engine to self-pack, self-arrange your cells, and just with a little bit of messing with numbers on some stem cells, you can generate some really, really cool organisms. And add some realism because there's some RNG involved in it. The physics engine, especially in Cell Lab, is not perfect. If you incubate, it is a lot less accurate. And if you do slow observe, it becomes far more accurate. So depending on the speed your game runs at, changes the reality of the math, or the accuracy of the math. So that can change the shape of the organism, how many of each organelle exist, the size of those Hox zones, and stuff like that. So you can get from just one design a large myriad of similar looking creatures that behave just slightly differently. Some might move fast, some might move slow, some might drift to the left, others might drift to the right, or have biases, things like that. If you had like eyeballs or brain cells being dictated by these Hox zones, you might have, through the RNG, one that can see better than others just because it has more sensocytes around its shell. You could even kind of mimic bacteria and have like more flagellum on the exterior or limbs sticking off and have that affect it. But there's a lot you can do. And I believe if more people continue to mess with these concepts in Cell Lab or other projects like Biospheres and Genomia and stuff like that, that we will find some very fascinating organisms develop through this method of mimicking reality using hormone or signal gradients. Is kind of just the best way. So I guess we can just call it like SG, signal gradients, as for the tech name, the technology name, or we can specify Hox Gene Expression Domains, even though this method doesn't really accurately represent what Hox genes are. In function it does, but Hox genes are very DNA based. So that, I think, that tech or that name is probably more accurately preserved for the players who are actually making DNA and RNA, like tape readers and stuff like that. So I think for the more basic players like me, we can probably just call it signal gradients or gradation or whatever. Maybe not, because coincidentally that's my initials, SG, and that seems a little egotistical, because I am not the first to mess with this. Again, I saw Aboba doing this, and but he was using it for a different purpose, and I got inspired by it. So I don't necessarily want this tech to be under my name. I am just showing you guys how it can be used. At the end of the day, I'm sure other players have discovered this in the past and then just never showed anyone, so we'll never actually know how original this is. I was just copying how I observed it in reality. So yeah, it was a very fun project to work on. I will continue to use it for fun. Right before we go, I guess I can just show you another organism that kind of uses it, like this guy. This is another one that I haven't uploaded a video on TikTok or anything about. So this one also does that. The way this guy does it is he has signal 1 produced on the left and signal 2 produced on the right, and you will see an outer ring form on his body, and it's color-coded of left versus right. And he does have a little bit of a brain. He's not programmed to do anything, though, so you won't see this guy do anything. But yeah, so very similar concept. You can create some very interesting shapes with this. Allow your imagination to go wild. In examples like these, this one does use cell splitting, like a max amount of splits. So this is more of an Enzyme and Kraken modded feature. So this one's only 7 long, so it creates a much smaller cavity. But it still works. On small scale, you can still get these distinct zones to appear, which is quite nice. And I believe in this example, I actually made them muscles on the outside instead of flagella. See? Very consistent. So this one formed a very consistent shell. So yeah, there's a lot that can be done with this. You can create some very interesting organisms. If we go to the absurd one, this is the maximum, because you can only go up to 20 with the max split feature. That might not be enough room. I do not know. But this one obviously has too many cells within its cavity, so it spits a couple outside. It is not very practical to do it this way. This is way too big. But yeah, it allows you to make like a variable length organism. It will adapt to wherever the secretion is being made, which is quite fascinating. This one developed very wonky. There's a reason I don't have this one as like a show-off piece, because it's too big. Sometimes it doesn't work. But yeah, so if this kind of stuff fascinates you, you can, continue watching the content. I plan on making more long-form content like this. I don't like editing. I'm lazy. I don't care for music. This is as raw as it gets, including me stuttering and spacing out and all that fun stuff. But for all the Cell Lab nerds or similar that want to learn more about this kind of stuff, I will continue making content about my more sophisticated or complex organisms. I don't know how much I will go in depth on tutorials, like, a whole long video of me programming every mode. Maybe someday I will make just like an hour-long video where I experiment and just make an organism off the top of my head so you can kind of see my creation process. Don't know if anyone would even watch that. My voice is quite annoying. So good luck making it through a video any longer than this if you even made it this far. So thanks for watching, anyone that did. This is like half an hour, so skim it, skip this part, I don't care. You guys have a good night.